Okay, we come to the last speaker of this session. He's Dave Farrens from Oregon Health and Science. And the title of his talk is Dynamics of G protein-coupled receptor activation and attenuation insights from fluorescence study. We will speak about also methodological aspects of fluorescence data.
Great. I'm going to start by analog timer. Make sure Terry's not pulling fast on me. Okay, for me it's a great pleasure and actually probably too much of an honor to be allowed to speak in this particular session, but I'll take what I can get. I thought I'd start out talking to you briefly about how several of the other speakers have mentioned, I think Wayne mentioned earlier about one of the happiest days of his life, or luckiest days, when he first got Govan to interact with him. And I had the same sort of situation, which I've never actually talked about this in public, but first I want to thank some people. Most of all, Dr. Karana. Govan was a great mentor, and one of the first speakers yesterday said it was one of the most intense periods of my life, and it was really fantastic for me, the training. I also want to thank Wayne, because my project involved a lot of collaboration between both of them, actually. So I kind of had the two-mentor thing going on. And finally, Dr. Rajmandari, one of the organizers. Tom has been a great guy to talk with and ask questions and co-mentor, if you will. And so I was a very lucky guy. So there's another guy I want to thank, and that's Nava Gupta. Some of you may know Nava from the Madison days. Nava was a professor of mine at the University of Nebraska, and I wouldn't be standing here if it wasn't for him. And here's why. This is a true story. I've never told this in public before. I did my undergraduate, I grew up in Nebraska, did my undergraduate degree in Nebraska, did my PhD in Nebraska. A lot of the time spent right here, working for Dr. Pilson Song, a great mentor, by the way, right around here in this place. So near the end of my PhD studies, I thought, well, I'd like to get into some postdoctoral research, and maybe get out of Nebraska. And so I sent out some letters. And back then, this is pre-internet days for the students out there, this involved putting your stuff in an envelope and taking it to the post office. This is the UNL post office, actually the City of Lincoln's post office. So I sent out some letters of query. And after a while, I got a couple of offers, but sadly none from Dr. Karana, where I really wanted to go. And after some thinking about this, I put a letter and some material in an envelope and sent it to one of these other people that showed some interest. And sadly went back to my lab and started one of these long preps we used to do back then. I worked on a plant photoreceptor at the time. And I was starting this project, kind of realizing I had committed to this thing, and I heard this noise coming down the hall, and it was Nava Gupta. And Nava had a very interesting way of walking. He kind of shuffled his feet, so I could tell it was him immediately. And he comes into the room where I'm working under these green lights, and he said, "Dave, I just got a phone call." Oops. And it's from Dr. Karana. He's interested in having you work for him. And I was sort of taken back by this, because I said, well, I just sent this stuff to the post office. My life is sort of set. And so he looked at me and said this, basically, Dr. And so the next words that came out of his mouth, I can still remember this, is, "You have to go get that envelope." I'm a Midwestern guy, and the federal post office is something you don't mess with. And so I was rather shocked by this concept. And I thought about the logistics. So this is a Google map, which we didn't have back then. There's a chemistry department at UNL. There's the post office. So somehow I had to get back there. And so I kind of figured out a pathway. By the way, there's the Huskers football stadium, in case you're wondering. That's the most famous place in Nebraska, sadly. So I rushed back there, got to the post office, begged these guys to give me this envelope back, and they did, actually. And so then I was able to contact Dr. Karana. So this hence began my first lesson with Govan, and the whole Karana family was Nothing is impossible, first of all. And there's a pattern to this method. And the first step usually involves this. You despair. And then you have to make it happen. That's just the way it is, right? And so it was really a wonderful experience for me. And thanks again, Go, for the opportunity. I'm just going to talk in the 2 minutes I have left here about some follow-up stuff that we've been doing in our lab. As Wayne Hubble mentioned, I got very lucky and kind of was at the right place at the right time with some of the spin labeling studies, and we were able to show with a group effort that there's a movement here of one of these helices when you activate it. This is a very old slide. Tom Sackmar's lab was also showing the same thing with metal binding sites, so sort of the field was converging, but the spin labeling data was very convincing. When I got my own lab, I decided to pursue this question with fluorescence. Well, this is just for historical reasons, the old model we put forth, sort of an early model movie, if you will. And all of these players contributed equally. Qi Yang enormously, was a great colleague to work with. And so when I got my own lab, I thought I'd kind of, for a long-term project, like to get into the question of does this helix move in other G-protein coupled receptors? Okay? And I also wanted to establish my own methodology. And so I basically copied the stuff that Wayne had done with EPR, or tried to develop site-directed fluorescence labeling. Because until this time, most people working with fluorescence were using different probes, but not taking one probe and standardizing it. But the idea being is you can measure mobility, accessibility, a number of these parameters with fluorescence. So the probe I stabilized on, and the one I'll talk about for a couple of the pieces of data I'd like to show you, is something called Biochemistry. Now this is a fluorescent probe that was first worked out by a gentleman named Ga C-terminus, a physical organic chemist, highly characterized this, and that was great to be able to go back to this body of data and look at its spectral properties. It's relatively small too. This is a key point Wayne being in my head about the spin labels. They're small, or minimally perturbing to the structure. Biochemistry is in that order of smallness, and it had been characterized also by Erwin London, a former Coroni. And so we set out to do something that to me was very inspired by the way Gauguin does things, is systematically characterize stuff. And so we went through T4 lysozyme. This is something Wayne Hubble had been doing with spin labels, and so we emulated that, put the Biochemistry probe at a number of sites, 21 total, characterized everything we could think of to measure, and now I had a data set to when I look at my probe, what do I read out? And we learned stuff about the solvent accessibility and some parameters if you're interested, but probably the most useful thing happened by accident for us. But because of the training I had with these wonderful folks, we were able to seize on that accident. And that is when we measured the fluorescence intensity, or quantum yield, in a couple sites we noticed it was very low. And every one of these sites that it was low was next to a tryptophan. All right? So then we could go back to this body of literature and find out that this probe Biochemistry is quenched by an electron transfer event, or proposed to be, by tryptophans to the Biochemistry. So now we had a built-in sensor, if you will, for low-resolution distance mapping. So we characterized this in a couple of papers with a great graduate student I had. But you see at places where they're the closest to C alpha distances, a substantial drop in the fluorescence intensity. And so this is very handy because you only have to put one probe. The tryptophan is always built into the sequence. So again, I would have never even thought to do any of this on my own, had I not had the fortune of training with both these guys. So now we can address the question, does Helix 6 move in other G-protein coupled receptors? And I also had the good fortune of hooking up with Brian Kadoka, who's an outstanding GPCR person, very dedicated scientist. He'd been doing some fluorescence studies. We decided to collaborate and use the Tryptophan method to look at ligand-dependent movements. And so what we found is that this movement, in the case of the beta-adrenergic receptor, only occurs upon agonist binding. There's no movement upon antagonist binding. In the case of Helix 6, at least that we could see with our methodology. All right, let me try this guy. Yeah, that's a little bright. Okay, so what we did, we used the exact same sites that had worked for spin labeling studies, and put the tryptophan in one position and attached the Biochemistry at another position. I had actually done this in my own lab with a graduate student, Jay Jans. We knew it would work for rhodopsin, so we thought, let's try it for the adrenergic receptor. Here's where those sites would be now on the actual crystal structure. And then this is where Brian Kadoka's vast expertise on working with that receptor came in so handy. He could now start scanning with different drugs, okay? And it makes for an elegant experimental system because you have the same labeled protein and all your variants of chemical that you put in. And you're looking for a change in the readout. So what we saw was no change in the controls, but a big fluorescent drop when we added an agonist. And for all these different agonists, we would see a fluorescent drop, no change with the antagonist. So this was kind of exciting. The guys in his lab pursued this further and were able to actually correlate radioactive binding, right, the KDs or the IC50s, with the fluorescence change. So everything was mapping quite well until you get to the catapult ring itself. And that's shown right here. In the case of the catapult ring by itself, even under saturating conditions, there was no fluorescence change up at the helix, suggesting to us that you need more to the ligand to induce this conformational change. I threw this slide in to suggest there's another GPCR where we've done this with. There's actually something called parapenoxin. We recently published this if you're interested in. What we found there was, this is almost all done by the Japanese group. They came to my lab a couple times. They have this interesting light. It's a rhodopsin family member that can activate G proteins, but it's about 20-fold less efficient than wild-type rhodopsin. And what they found through their studies is, at least by our assays, there's not as large a movement, all right? Suggesting that movement may correlate with this ability, or the efficacy that Tom mentioned in G protein activation. I also want to make sure I mention something right here. This guy, Tsukamoto, for any of my Japanese colleagues, he's an outstanding young postdoc that's right now looking for a job. In fact, at the moment, he's interviewing somewhere. So if you have any openings, this guy is really fantastic. He's been in my lab 6 months, already written a paper, left it with me before he left. He's just that kind of guy. So I would highly encourage anybody to contact him or me through him. I hate to lose him, but he deserves better than my lab. I'm probably joking. So, further directions we're going with this, Tom mentioned, Sakmar, about the whole idea of inverse agonist, partial agonist is something that Brian Cook is actively pursuing, and we're doing some to help him there. We have some interesting results where the amount of change we see can relate to how effective or efficacious the ligand is, even under saturating conditions. So that's kind of exciting. I'm not going to talk about that right now. I'm going to finish this up to get after something that soon after the helix movement stuff came out, I got kind of interested in the question of why would this helix move? What would be a role for this? Here's one joke to get to the other side, but that only buys you 30 seconds in a talk, and then you still have to get back to, this is the hypothesis that's been put out there before us. Simon Karnick years ago, several years before we did some work, had suggested this from some mutagenesis studies. Other folks had proposed it. We set out to try to measure this, all right? And so this is the region that Wayne discussed, this extreme C-terminus of the alpha subunit has been known to adopt the structure in the NMR studies upon binding. It seemed to be disordered, but no one really knew that. And so, I just learned that from Wayne, actually. The approach was, let's go to the surface of rhodopsin now and attach binding probes at several different sites, okay? And see if we simply, this was one of those rare experiments where you think about it and it actually works start to finish 4 months. I mean, it's amazing for us. That never happens in my lab. So we ordered up some of this peptide with a tryptophan at the end. And the experiment was very simple. Add in the C-terminus and look for fluorescence quenching at any of these sites. By the way, Jay Jans, the guy who did this, was my first grad student. There's an interesting history for him. He was an undergraduate student here at Wisconsin, worked with Julius Adler, right? Then came to work for Mark Krebs for a couple of years here at Wisconsin. He came to my lab as a PhD student and went to Tom Sakmar's lab. So he's really an outstanding guy, really, I mean, quite a producer. Anyway, he was able to show that really only this region would show any quenching upon excess of the tryptophan containing peptide, and a cysteine right here was protected upon binding, okay? So we came up with this theory that there was a hydrophobic patch that this C-terminus was sensing and binding. And this is something, again, I must give credit to Wayne for, because I would have never thought of this experiment except for some conversations I had with him when I was a postdoc. As we put in extra mutations in the receptor, what we would see is, let me back up. We put some mutations we knew from the early spin labeling days that would block G protein activation. But we never knew if that was because the G protein couldn't bind or if it was because it couldn't activate. Those are two different questions, right? With this peptide assay, what we could do is go in and add more and more peptide to see if we finally saw some binding, right, some quenching. And the reason that I'm crediting Wayne for this idea was he pointed out to me that biochemists never really think in thermodynamic senses. You can have a few kcal per mole change in the free energy, but that's an enormous difference in concentration. And nobody in their right mind would throw 2 or 10 millimolar transducin on something to see if it would bind. But we could do that with these mutants, and what we found was, indeed, these mutations would kill G protein activation were actually blocking binding, but if you put enough peptide in, they could still bind from the C-terminus. So that was kind of you can actually calculate kcals per mole because you had 2 different concentrations. Hence our conclusion that this hydrophobic site provides some kind of binding affinity for the C-terminus of the G protein when it binds. And we were very happy to see last year some wonderful crystal structures came out, and indeed the C-terminus does bind in where we roughly predicted. We actually had a model that was pretty close to this, but I was too chicken to publish it because I didn't think our data really was worth it. But anyway, that does seem to be where the C-terminus binds. So to answer that question, why is the C-terminus, why does this helix move? We think it's to allow this part of the G protein to bind. So let's answer that. I should really delete 5, but that's more from the crystal structure. There's clearly a movement at helix 6 upon activation. In other GPCRs, it's not nearly as well known as rhodopsin, so there's a lot of room for future research. And why does this movement occur? To enable binding of the C-terminus. So with that, I will end. Do I have 4 minutes left? 3. Okay, I want to show you one last thing. And then we'll break. Because this is a new thing that we're developing in our lab, something called, which we've basically taken the old thing and put a new acronym on it. All right? I've learned from the market people. And now I have a sexy acronym, TRIC. Tryptophan induced quenching, or TRIC fluorescence, or if you revert it, you have the fluorescence TRIC. And what we found is a new way to calculate exactly how much of this quenching is happening by the probe and the quencher being in contact and how much are not through a dynamic event. And I'll try to get to these 3 questions really quickly. This came out of some work where we were trying a number of different probes. This is one that Ulrike Alexia, who's here, has been using, Lucifer Yellow, binding, several other ones. It's essentially redo the calibration for probes that span out all the way into the red region for fluorescence studies. What we see, if we compare this as a function of distance, is the farther out you get, the less quenching you get before and after having the tryptophan there, okay? For some of these longer probes, they're very efficient quenchers. What we were seeing, though, is that there's no correlation with the change in the lifetime. So for a quick crash course in fluorescence methodology, theoretically, if there's a change in intensity, there should also be a change in the rate of decay. They should mirror each other, right? A de-excitation event. You can run into situations where you can have a huge drop in intensity with very little change in the lifetime. The y-axis here is lifetime in these different positions. So the interpretation of that is that they form some kind of static complex at the moment of excitation. And I'll show you what I mean by that. Here would be a situation where the fluorophore is in an open situation. Okay? So you excite it with light, it can either fluoresce, unquenched, or perhaps there's this electron hopping event to reduce its fluorescence. Both of these are dynamic events. There's a whole other category where if the tryptophan is touching, or any quenching group, the fluorophore, upon light excitation, it never becomes fluorescent to begin with. So if you sum this, you'd see a bigger drop in the fluorescence than you would see from monitoring this event, which would be the change in the lifetime. Okay? Why is this? This is the who care parts. I've never been able to figure out how to calculate the amount of that static amount. Okay? Because I always would get hung up on this part. How do you calculate the concentration of the quencher? It's infinite. It's there every time. Right? And I dug through some literature and found a thesis from 1970 from Gregorio Weber's lab, a guy named Richard Spencer, and they ended up doing studies of flavin adenine dinucleotide, which should be fluorescent, but it's not, because the quenching event, this acts as a quencher. And these guys had proposed that it was actually stacking, right, forming stacked complexes. Well, for me, this is the same concept, except I have a protein instead of this linker, right? So I got this guy's thesis, and that's a whole other story that you don't even want to hear about because we don't have time, and it was more difficult than it should have been. And I'm going through 300 pages of this stuff. You don't see theses like this anymore, by the way, students, so just keep that in mind. Typewritten, 300 pages of derivations. Quencher there every time. I finally get to this sentence. The intermolecular quenching, intramolecular, is concentration independent, of course. So it's a monomolecular reaction, you just throw out the quencher concentration. So that's one of those classic, oh, God, wish I thought. It really wasn't that hard. And so now what we can do is you can determine the amount that's open by simply the ratio of the steady state change over the lifetime changes, and the amount that is in this complex is simply 1 minus that. So now we can measure the amount that are in these static complexes, and this is something we've been doing with Brian Kabilka to look at seeing changes. And so I'm going to stop right now, Parrot. But the point of that is this isn't the kind of elegant distance that one could say of 1 or 2 angstroms, but if two things are touching each other, they're by definition close. Is the way I would make this argument, right? And if 50% of them are close and they go to 10%, there's been a change in that spot. So I'll end with that, and thank you.